Welcome. We're a Korea-based biotech company, Celebrate Therapeutics. We are delighted to have this opportunity to be able to present our recent research on our novel COVID-19 therapeutic, improved cell-permeable nuclear import inhibitor, in short, ICP-NI. Using Celebrate's proprietary delivery technology, namely therapeutic molecule systemic delivery technology, we have developed an innate immunity regulating, antiviral, anti-inflammatory immunotherapeutic agent ICP-NI as a COVID-19 therapeutic. Our first slide shows that intracellular delivery of nuclear localization signal suppresses COVID-19 mediated inflammation by regulating expression of cytokines and chemokines. ICP-NI is composed of cyclic nuclear localization signal fused with Celebrate's novel cell-penetrating peptide. When our body experiences foreign substances, such as SARS-CoV-2, coronavirus, our body activates innate immunity, such as stress-responsive transcription factors, such as NF-kappa B, NFAT, STAT1, and AP1, which binds to importin alpha for nuclear translocation, producing cytokine as a defense mechanism. When an unknown substance is introduced, our body overly produces cytokines, and this phenomenon is called cytokine storm. Cytokine storm is accompanied by systemic inflammation, multi-organ dysfunction, disseminated intravascular coagulation, fibrosis, septic shock, and even death, which are the symptoms observed in COVID-19 patients. As a development process, we examine cell permeability and activity of ICP-NI. In Figure C, ICP-NI showed substantially increased cell permeability compared to its control. Moreover, we confirmed that ICP-NI blocks nuclear translocation of SRTFs by confocal microscopy, Figure D, and Western blot analysis, Figures E, F, and G. The next slide shows that ICP-NI suppresses immune cell homing, cytokine storm, and tissue inflammation in poly-IC inhalation-induced pneumonitis animals. In Figure A, ICP-NI suppressed homing immune cells 2 hours after challenge by 82% in bronchoalveolar lavage fluid. Moreover, ICP-NI decreased pro-inflammatory cytokines such as interleukin-12, TNF-alpha, interferon gamma, MCP1, and IL-6. On the other hand, ICP-NI increased anti-inflammatory cytokines such as interleukin-10. On H&E staining, we were able to see that ICP-NI prevented pneumonitis and recovered porous structure. The next slide shows that ICP-NI protects lung tissue from pulmonary fibrosis in intratracheally challenged bleomycin-induced fibrosis animals. In Figure A, micro-CT data shows that ICP-NI protects lung tissue from pulmonary fibrosis. As you can see in the yellow circle, ICP-NI prevented the formation of white pulmonary fibrosis. And in Figure B, the quantification shows that the degree of reduction was 61.3%. In Figure C, the serous red staining has shown that ICP-NI not only prevented parenchymal collagen formation, but also helped to maintain normal porous structure, which suggests that ICP-NI successfully blocked pulmonary fibrosis. The next slide shows ICP-NI protects immune cell death caused by systemic inflammation in poly-IC SCV-induced pneumonitis animals. In Figure A, ICP-NI blocked pneumonitis-induced apoptosis of splenocytes, and this observation was reconfirmed by apoptotic staining in Figure B. In Figure C, we checked the population of individual immune cells in spleen, such as total T cells, CD4 positive T cells, total B cells, and macrophages. These results show that ICP-NI protected functionally active immune cells by suppressing cytokine storm. Our next slide demonstrates that iCP recovers clinical symptoms in SARS-CoV-2 infected non-human primates. The efficacy of iCP against COVID-19 was examined in SARS-CoV-2 infected non-human primate models using a single injection protocol shown in Figure A. iCP recovered critical clinical symptoms such as O2 saturation, respiratory rate, body temperature, and glucose level in fully established COVID-19 monkey models. Our final slide shows that iCP recovers clinical symptoms and also decreases viral copy number in SARS-CoV-2 infected non-human primates. In Figure A, iCP even recovered clinical symptoms in partially established COVID-19 models. For instance, O2 saturation was recovered by 120%, respiratory rate was recovered by 50%, and the heart rate by 67%. More importantly, iCP decreased the viral copy number by 82.4% in both fully and partially established COVID-19 non-human primate models 5 days after the challenge. In conclusion, iCP by suppressing cytokine storm protects immune cells, and by this mechanism, iCP projects antiviral activity. This suggests that iCP is an innate immunity-regulating antiviral, anti-inflammatory immunotherapeutic agent which has a great potential as a COVID-19 therapeutic. Thank you for listening. If you have any questions, feel free to email us.